Greetings, friends. Welcome back from spring break. I don't know about you guys, but I sure enjoyed that week off. And I've been having a great time with my family. The kids are getting along really well together. And things are actually going pretty good. I hope they're going pretty good for you guys too. Looks like we're going to be doing this the rest of the year. Unfortunately, that's not something I'm stoked on because I do like spending time with you guys. But we'll just make the best of it. We're going to do this COVID-19 briefing for 2 more days only. And that's because biotech kids need to start getting ready for their industry certification exam. And chemistry kids need to start learning about acids and bases. So we'll just do this for 2 more days and we'll pick up where we left off and kind of change what we were going to do a little bit. So, COVID-19 pandemic briefing brought to you by Pirate Biotechnology, Morro Bay High School. So here's what we originally thought that we were going to cover. We originally thought that we were going to look at what everyone's freaking out about, which is epidemiology, which we did do. And then we went on to learn what coronavirus even is. That's the study of virology. And then we looked at how coronavirus causes COVID-19 disease, and that's what's called pathophysiology. So what we were going to do next was we were going to look at what sort of medicines might help in the treatment of COVID-19. And then we were going to look at, what a vaccine is and how a vaccine works. We're not going to do those next 2 things. That would take another couple weeks. So instead, what we're going to do is something that we need to do anyway, which is important, which is study why testing is important and how the testing works. So part 4, why is testing important and how does it work? This is the study of diagnostics. So why test even? Why even test? Here's why. Remember back to this model right here when we were studying epidemiology. We looked at 4 hypothetical models of how disease would spread through a population. We looked at a free-for-all. We looked at where a quarantine was attempted to be applied to a certain population. We looked at moderate distancing and its effect, and we looked at extensive distancing and its effect. And there's 3 colors here, and the light blue color, that represented susceptible people that are susceptible to the illness. The brown color represents people that are infected, and the pink color represents people that were infected. So in order to understand it, just think of yourself as a pixel right here, and you're a blue pixel. You're a susceptible person. You have not gotten the disease. And then think about this X timeline, or this X-axis is a timeline. So here you are. Just think of yourself as this blue pixel, you're cool, everything's fine, boom, you're infected. You remain infectious throughout all this time, and then bink, you recover or you die. Most of the time you recover. So once you're recovered. And what we're thinking right now is you cannot get this same coronavirus again once you've recovered, nor can you pass it on to anybody else. So here's these 4 different, what we need the population to be purple. We need the population to not be infectious and not be susceptible. And in order for that to happen, one of 2 things needs to happen to most of the population. Either 1, everybody needs to get it, or almost everybody needs to get it, or 2, we need to develop a vaccine. And a vaccine is not coming for, they say, at least a year, and it could be longer, and it could be never. So that leads us to the other thing that we would need to do, which would be to get everybody sick that is not at a higher risk of developing ARDS and getting really sick and getting permanent damage or dying. So that's what we're talking about right now, because if you keep everybody distanced, it just keeps dragging on and on and on forever. And meanwhile, the economy is suffocating. So here's where we are as of, I guess this would be yesterday. And this is in the United States. And you can see that we've succeeded so far. We have been distancing, and that has caused the curve to bend and flatten. So that seems like great news. But the thing is, we might be, oh man, how do you get out of this thing? We might just be right here. See this little place right here? It looks like it flattened out and it looks like it's going down, but it could still go up more. It could still drag on for a long, long time. So although we succeeded in flattening the curve and prevented our hospitals from getting overrun, that was our mission. That's what we need to do. That's what we needed to do acutely right away. But we got that done. We need to move on to the next phase, which is trying to develop what's called herd immunity. So once the majority of the population is purple in this model right here, which means that they have recovered, then the rest of the people, the old people, the sick people that you do not want to get COVID-19, are safe. That's what we want to achieve. And in order to achieve that, we still need to get our testing or our diagnostics capabilities up and running. So there's two categories of tests for coronavirus. There's tests to determine if you have coronavirus, and there's also tests to determine if you've had coronavirus. And both of them are equally important. So the test to determine if you have coronavirus, that's important to prevent you from contacting people who have not had it. So to identify, yes, you have it, put yourself in quarantine, don't go around anybody until you don't have it. That's why that's important. Also, you can contact trace, so you can look up a history of contacts you've had with other people, and government agencies can call those people to warn them that they have been around somebody that has coronavirus. So that's why that test is important. And that to test if you have coronavirus, you do something called RT-PCR. And the RT stands for either real-time or reverse transcriptase polymerase chain reaction, RT-PCR. In this case, it stands for both real-time and reverse transcriptase. Don't worry about it. I'll tell you all about it. The next test is a test to determine if you have had coronavirus. And that test is done with something called ELISA, which is an acronym for something like enzyme-linked immunosorbent assay, something like that. And I'll tell you guys all about that too on Friday. So today what we're going to talk about is this RT-PCR, which is a test to determine if somebody has coronavirus right now. So in order to understand how it works, we're going to start with the central dogma of molecular biology, which sounds really gnarly and confusing, but don't worry because you learned all about it in biology class. This is the idea where you take DNA, which is instructions, and through a process called transcription, cells make an RNA copy of that DNA, and then through a process called translation, cells take that messenger RNA and they use its instruction to link up a bunch of amino acids in a certain sequence, which folds into a certain shape, which allows it to do a certain function. And we call that item a protein. So that's what normally goes on inside of cells. And the reason I'm telling you this right now is because it's important for you to understand how this test works. So the enzyme that does this step, which is make an RNA copy from DNA, that's an enzyme that's called RNA polymerase. And these words look really confusing at first, but later on you learn that it tells you in the name what it does. So to polymerize means to make something. ACE means an enzyme, and RNA is the thing that this enzyme is polymerizing. So there's an enzyme called RNA polymerase that does transcription. Better act, somebody. Okay, there's another enzyme called reverse transcriptase. And this isn't exactly true, but human cells do not have reverse transcriptase. Reverse transcriptase comes from viruses called retroviruses. Coronavirus is not a retrovirus, but HIV is a retrovirus. So I'll talk about how it works in a second. First, I want to talk about what it does. So what reverse transcriptase does, which is an enzyme, what the enzyme reverse transcriptase does is it does reverse transcription. And what reverse transcription is, it's the opposite of translation. It takes RNA and it uses this enzyme in order to make DNA. So it does the opposite of translation. What's the opposite? It does the opposite of transcription. What is the opposite of transcription? Oh, you got that right. It's reverse transcription. Oh, they gave it a good name. Here's the deal. Here's HIV right here, or some other retrovirus. What it does is it binds to a host cell, one of your cells, and the virus enters and it opens up and it releases all this good stuff in here. The good stuff is viral RNA, just like coronavirus. But unlike coronavirus, what happens is this viral RNA gets copied into DNA by an enzyme called reverse transcriptase. And then another enzyme goes and it splices that DNA into your DNA. So now your DNA in your cells contains all the instructions for how to make HIV. And that's what your cells do. They make HIV, and the HIV explodes from your cells and infects other cells. And that's how retroviruses like HIV work. The reason it's important for us to talk about it right now is because of this enzyme right here, reverse transcriptase, is a tool that we're going to use to test somebody to see if they have coronavirus or not. We're going to use this enzyme tool called reverse transcriptase. All right, so here's what happens. You guys have seen this in the news where they take the swab and the person's like, oh, because it's super uncomfortable because they have to take that swab and put it in your nose way back here and tickle the back of your throat with it. They're starting to develop other methods of sampling where you don't have to do this, but this is the surefire way to make sure that you're getting some sample of virus if it's there. So once you get that on the swab from the patient, you put it in this tube right here, which contains a solution which protects any viral RNA, because you don't want that viral RNA to get all chewed up because you're trying to test for it. And if it gets chewed up by some enzymes, some other enzymes or something, then your test is going to come out negative and it's going to be a false negative. You don't want false negative tests. Talk about that later. Okay. Step one, don't worry. What I mean is, honestly, don't worry about all these details right here. This is not enough time for me to go over all this with you guys. I just want to give you the idea of how this works. So you take that sample, and what you do is all these steps. This is somebody working in a lab with a pipette and with tubes and with all these tools, and they're performing all these steps in order to purify the RNA from that sample that was taken off of that swab, because there's all kinds of stuff on that sample. There's like mucus, and there's cells from your body, and there's maybe virus cells, and all those cells have hundreds or thousands of proteins, fats, DNA, RNA, all that stuff. And you need to separate the RNA from all that other stuff. And it's time consuming and it requires all of these tools and it needs to be done right or it won't work. Okay, once that's done, here we are right here. We got our DNA extracted. Once we extract that DNA, we're going to do reverse transcription on it, which is going to turn it, we're going to turn the RNA into DNA. What tool are we going to use to turn the RNA into DNA? I'll give you a hint. It's an enzyme. Oh, you got that right. We're going to use reverse transcriptase. So we're going to take some reverse transcriptase, we're going to put it inside this tube right here, and that's going to make a bunch of DNA for us. Then we're going to take that DNA and we're going to dilute it, and then we're going to put it into these wells right here, and we're going to add some other stuff to it to make a whole bunch of copies of the DNA, which was made from the viral RNA, if any viral RNA was there. So in order to show what's going on inside of that tube, I thought I'd show you guys this thing right here. Let's check it out. And they're not going to put any sound in here. This is a nice model right here. So here we are with your tube. You put reverse transcriptase enzyme in there and you put RNA in there. Maybe that some of that RNA contains viral RNA from coronavirus. So what happens? Oh man, I wanted to pause that sucker. So what happens is a bunch of copies of that viral RNA get turned into DNA by this enzyme called reverse transcriptase. Then you take it and you put it into another tube and you use another enzyme called DNA polymerase, which the biotech kids are going to know all about. And you do something called PCR, which makes a buttload of copies of that viral DNA. So we won't go over how that happens. Just know that what we did is we took a whole bunch of, we took a little bit of viral RNA and we turned it into a buttload of viral DNA. But it only works if the viral RNA is there in the first place. And the reason we can make sure that it only works is because we use these things called primers, which you'll learn all about in biotechnology. You biotech kids have already learned about it. By the way, in biotechnology class right now, we would be doing RT-PCR in class this week. I'm a little bit sad that we're not. Actually, I'm way sad that we're not, but it's okay. We're just going to carry on. So here's what the results would look like. Let me go back here a little bit. So something else you put in this tube right here is this fluorescent molecule that only sticks to double-stranded DNA. And the more double-stranded DNA is in there, then the more that this fluorescent molecule is going to light up. This machine right here does PCR, but it also contains a spectrophotometer, which can measure how much light is being released by the tubes. And the more DNA there is in the tubes, the more light is going to be produced. So that spectrophotometer ends up showing a graph like this. And this is called the threshold right here, which is a level of light that means that something's going on. Anything less than that means nothing's going on. This right here is what a positive control would look like. So a positive control means that a researcher put some coronavirus in the tube to make sure that it's working, to make sure that the procedure's working. This would be a positive sample right here. You can see that it goes right along with the positive control. This would be a negative control right here, and this would be a negative sample. Remember, this is how much light is being emitted right here. So that's how reverse transcriptase PCR works. And I think now you guys can maybe understand a little bit better why we've been having so much trouble getting this testing regime up and running, because it is not kindergarten. We could do it in the lab. We could do it in the lab, but we'd probably mess up a bunch of times. We'd probably get some false positive results sometimes, probably get some false negative results sometimes. And they can't have that. It has to be nails. So in order to make it nails, every lab that's doing these tests needs to be certified to show that they're super bad. So that's it, you friends. I love you guys so much. I can't wait to see you again. Sorry this video was so long. Oh.